Hi everyone, my name is Aditi, and I'm joined here today with my co-authors on this project, Daniel, Ben, and Lucas, to go through how to use and run Proto for biological design.
Proto is a high-level programming language and tool platform for generative biology. The goal of Proto is to make it easier to compose biological AI models into design workflows without needing to rebuild a custom pipeline for every new task. Many biological design campaigns depend on a combination of sequence generators, predictive models, scoring functions, optimization algorithms, and downstream analysis tools. But these components are often difficult to install, configure, and connect. Proto provides a shared framework for bringing these tools together using 4 core primitives: sequences, constraints, generators, and optimizers.
When you open the Prototypes tab, you can see your existing designs. You can also use pre-built example programs. In this example, we will create our own. To start, we'll take a look at the Proto canvas, which is the graphical interface for building Proto programs. The canvas lets you work with the same 4 core pieces of Proto: sequences, constraints, generators, and optimizers, but as visual blocks that you can configure and connect. This makes it easier to see the structure of a design workflow: what sequence you're designing, what tools are scoring it, what model is generating candidates, and how the optimizer is guiding the search. For this demo, we'll build a simple promoter design workflow directly in the Proto canvas. The first primitive in Proto is a sequence. This is the biological sequence we want to design or modify. In this case, we'll use a DNA sequence because our goal is to design a short E. coli promoter. To start, I'll start by dragging a DNA sequence box onto the canvas. This block represents the candidate promoter sequence that the rest of the workflow will operate on. Within this block, you can do things like change the length, rename the segment, and specify an initial sequence to start with. For this example, we'll use a random sequence to start. The next primitive is a constraint. A constraint is a scoring function that tells Proto whether a candidate sequence has the properties we want. For promoter design, we care about whether the sequence looks promoter-like and whether it might be likely to drive transcription. For the purpose of this simple demo, I'll stick to adding basic constraints, like the presence of a -35 and -10 sigma 70 box motifs, and the basic sequence identity metrics like GC content. Each constraint gives Proto one signal about what makes a good promoter. The third primitive is a generator. A generator proposes new candidate sequences for Proto to evaluate. Here, I'll use random nucleotide mutation as a simple generator. Random nucleotide mutation makes small random edits to the DNA sequence, which is useful for this demo because we can start from a randomly generated DNA sequence and explore variants till we converge on something that best meets our constraints. The final primitive is an optimizer. The optimizer coordinates the design loop. It takes candidates from the generator, evaluates them with constraints, and decides which candidates to keep. For this example, I'll use MCMC as the optimizer. MCMC is a good fit here because it can accept or reject mutations based on the combined constraint score, allowing the sequence to gradually move towards better promoter candidates while still exploring the candidate sequence space. Once these blocks are on the canvas, I'll connect them together. The DNA sequence is modified by the uniform mutation generator, scored by the promoter-related constraints, and optimized by MCMC. Then we can run this workflow and have Proto search for promoter sequences that better satisfy the design objective. As you can see, the program is now running and generating an output energy graph demonstrating how the designs are converging to an optimum over the number of iterations we specified. In addition to allowing the program to run, you can also view sequence output metadata for each of the generated candidates and inspect the outputs to determine how well they comply with your constraints.
To assist with designing new programs in the front-end interface, users can open the Proto chat assistant on the right. This agent acts as an educational resource as well as a design co-pilot to guide users through the Proto language user interface. For example, the chat can help a user build a program from a biological design specification, discover appropriate constraints, generators, and optimizers for a desired task, fetch real biological data from public databases like the PDB, UniProt, NCBI, and provide scientific guidance on open-ended design tasks or questions. Let's see it in action by asking the chat to design a basic toy example. Our example prompt is, create a 500 base pair DNA sequence with 70 to 80% GC content and no homopolymers longer than 4 bases. The agent then reasons and explores our existing catalog of constraints, generators, optimizers, and example programs to identify the appropriate components required to assemble the program. As the agent reasons, users can inspect the reasoning traces and tool calls of the assistant to get a deeper understanding of how the agent gets to its final program design. Finally, the agent generates a program in the format that we expect. You can then import and visualize the program onto the canvas. If necessary, you can also tune the parameters or simply compile and run the program.
To empower the programming layer Aditi and Dan just showed you, we needed to build a way to quickly run and compose a wide variety of tools and models. In addition to the optimization framework, we are also releasing Proto Tools, an open source codebase containing ready-to-run local implementations of some of the most commonly utilized biological models. With one pip install command, scientists gain access to a massive range of tools, including biological language models, structure predictors, inverse folding, sequence analysis, gene annotation, conformational dynamics, and genomic scoring, and more. Under the hood, every tool runs in its own automatically managed, isolated environment, so all dependency wrangling is handled for you. Proto detects your GPU and drivers to install versions of the tools that actually work with your hardware. And because everything is managed separately, you can run all of the tools from the same codebase. In addition, Pro Tools implements extensive infrastructure for features such as device management and GPU fanout, making it easy to call tools in quick succession. We've linked our doc page here so you can learn more.
To make these tools even more accessible, we've also developed a cloud-hosted backend that is reachable by the public through a graphical user interface on our website or through an MCP server built for interaction with coding agents. In this tutorial, we'll focus on interacting with Proto Tools through our website, where you will find a scrollable list of cards for each of the hosted tools we currently support. To sort through the catalog, you can filter by category to see the full design stack. Or, if you already know what you want, just search by name. Let's see one run end to end. Let's go through a quick demo of the graphical interface for Protein MPNN. Protein MPNN is an inverse folding model. Given a fixed three-dimensional protein backbone, it can predict amino acid sequences that are likely to fold into that shape. Let's use Protein MPNN to sample some sequences that are likely to fold into the shape of green fluorescent protein, otherwise known as GFP. First, we need to specify the structure of GFP as an input. Here I will do this as a PDB file I downloaded from the AlphaFold database. After we have uploaded the structure, we can visualize it in the interface. Before we start sampling new sequences, there's one thing we want to set up. Right in the center of the GFP barrel, 3 residues form its chromophore, the part of the protein that actually absorbs and emits light. In wild-type GFP, these are residues serine 65, tyrosine 66, and glycine 67. We don't want protein MPNN to touch them because their identities are essential to the protein's function. So, rather than redesigning them, we will mark them as fixed positions, using the field and specifying the range of 65 to 67 as fixed. Protein MPNN will then redesign every other residue around them while holding these 3 exactly as they are. We can also change any configuration settings that we can change on the Pythonic layer of the tool's codebase. In this case, let's change the number of sequences we want protein MPNN to return to us to 20. Then we can click Run. Once the tool has run to completion, we see our expected output. In pink, we see all the amino acids that were newly designed, with the positions we fixed made visually distinct from them. Every tool hosted by Proto has curated input and output renderers to facilitate parsing information at a glance. And that is Proto. Everything we've shown to you is open and available to everyone. We hope it makes biological design more composable and accessible, so that science continues to be the centerpiece, not the setup. We're very excited to put this in the community's hands and can't wait to see what you build with it.